Raleigh Hood UMCs. I just got back from a month-long cruise in the eastern Indian Ocean where we sampled the marine food web all the way from tiny phytoplankton up to small fish, from 40 south all the way up to Jakarta. And I was amazed. I was blown away by the changes in diversity along that transect. We saw at the far southern part of the transect, the grazers appeared to be dominated by pteropod larvae, okay? They just were eating all the chlorophyll. And then by the time we got up near Jakarta, the grazers were dominated by tiny little zooplankton that we could barely see. And my question for you is, can these relationships and models, and of course I'm a modeler, so this is kind of mean maybe, but can these relationships capture that? I mean, and it's not just the size structure that's changing, a complete reorganization of the food web. So can these relationships deal with that? Are they capturing that?
So I think one of the questions is, are they capturing it explicitly or are they capturing it implicitly?
Yes.
Right? And so one of the attractions about doing things like allometric scaling is that you might be able to capture things implicitly that you're not capturing explicitly. And at some level, things like the Epley curve, the whole idea of the Epley curve is taking a known diversity in plankton and just saying, well, the succession is going to be handled by the fact that as I move to higher temperatures, something that is more adapted to that temperature will fill in from something that's less adapted to that temperature. And so intrinsically, I think there's no reason to think that you can't, but there's also no reason to think that you can without trying it.
Comment on that as well. I mean, I guess it's also a question about thinking what are the main traits that are changing, not necessarily the main species, but if there's a fundamental trait axis that you can use to describe that variation that may inspire you to which of those traits that you would want to choose to capture those changes.
Hi, I'm Priscilla Lang from the OCB, NASA. I have just a quick question about the physiological parameters that you were using in your model. Does it incorporate, or can it incorporate symbiosis? Because apparently in some of the provinces, symbiosis is quite important, and it depends a lot on the nutrient supply and the type of nutrients that is there. So can it be incorporated in some of the regions of the ocean?
Yeah. Actually, I would think that when you have a symbiosis, for instance, between a diatom and a diazotroph, you could also think of that as, just one organism, that they're intrinsically connected. So you would write the model of the generalist cell saying, well, I had up there with the different feeding modes that they could have, and add diazotrophy in there. And in that way see, well, under which cases would diazotrophy come into it as well. And if that happens together with phototrophy, well, you can interpret that as a symbiosis that's happening. You wouldn't model the actual act of symbiosis or the mechanisms that's stabilizing the symbiosis, but you'll just model the function of it. If it just changes a little bit, it doesn't really matter so much. What you have to think about is where these changes in size matters is when it's on big scales.
Hi.
Hello, this is Brendan Carter from the University of Washington. So it seems like one of the kind of tensions that I'm hearing in the conversation right now is that some folks are like interested in a specific mechanism because that's the focal thing that they study, whereas it seems like one of the principles of using this allometric approach is that you're kind of glossing over a lot of that complexity. And that might be appropriate when your goal is to push nutrients around in a model. So I guess I'm curious, somewhere between kind of detailed ecosystem exploration and mechanism and biogeochemical modeling of specifically just the chemicals, where would you say the allometric approach kind of falls in there? What things is it appropriate for looking at, and what things is it kind of smoothing over too much?
You're looking at me. On an ecosystem level, I think that it's to me the strength of it is there's just a pattern, and so the question is like the pattern is just it's an observation. So the question is what's underlying that pattern, and so I think that goes back to the idea again of the deviations are probably more interesting than the line itself or the that trend itself. So, you know, I think like as an approach, like when can you gloss over the things? It probably depends if you care about the details or you just want kind of this macro emergent thing. And I think that's probably true on any of these cellular levels too. Of like, you know, do you need a mechanistic thing or do you just need something is there a pattern? And if so, then you could use that.
I would also say that you can use the allometric relationships as methods of data reduction, and we need methods of data reduction, right? But by definition, when you do data reduction, you are throwing out data.
I can add to that a little bit. I also think sometimes we're interested in variability within subsets of species. So we're talking about global models and modeling whole ecosystems, but sometimes it's important to focus in on one subset. And so I think allometric scaling works really well, especially when you're looking at a subset of species or maybe even one location or something where you take out a lot of that variability just by narrowing your study and sometimes learning from those narrower studies.
Hi, Laura Holland, University of Rhode Island. I'm a PhD student, and I don't know anything about modeling, so I'm just going to preface that. And I don't know if this is a silly question, but Ken, I was wondering, this allometric scaling approach that you use with the traits, I think it's really interesting because you're getting at that diversity, you know, between the functional groups and within the functional groups. But I'm also wondering, how do you think this compares to like a gene-based model, like the genome model, I think?
Yeah.
How do you think it would compare? And do you think in the future we could maybe combine the two approaches? Because I think that would be great. But
I think the two approaches are very similar, actually. It's just a question of words. I mean, you can call them genes or you can call them traits, but by and large, that's more a question of semantics. I think what the genome thing does is we're trying to map it directly onto trait, and I think that's a great aspiration, and we can do that for some of these things because we can actually measure the expression of the traits. A little bit like I did in the size-based model, I looked at, well, how much was they getting from phototrophy. You can probably measure that directly from a metagenomic, is how many RNA are expressed that's related to photosynthesis, and the same with nitrate uptake, et cetera, et cetera. And doing that mapping is great, and I think that's something we should do. I think it gets harder when we get into more complicated organisms, eukaryotes even, because then it gets just messier with the genes and what they do. Just measuring genes that codes for phagotrophy is hard.
Ken Johnson, Mabari. The question I'd ask is what's the future of scaling allometry to behavior? And the example I've just been shown recently, Kelly Benoit Bird is running a sounder at 1,000 meters depth off Mabari, looking at vertical migration. And if normally she sees not much migration, if you see anchovies show up in the system, it just turns on, you know, intense migration. And if you see porpoises show up in the system, the migration goes deeper. So, you know, what's the future of that?
I think that, so that, I mean, allometry of course come into that, but only because of the bigger ones are eating the smaller ones. But the future of that is, I think a little bit like Karen showed, we can model that mechanistically now if we combine with game theory, and we can even do it with several trophic levels and see how they interact. And beginning to pretty well actually describe the type of pattern also that Karen showed, that Mark Roman had with size and the different depths. We can understand that also actually mechanistically by combining these allometric relationships with the game theory, the evolutionary game that they play. So I think that's the future of it. At the moment, from a modeling perspective, it's fairly heavy. So the challenge now is to develop methods that simplifies that to the degree that we can put them on our regional or our global models. I think it's absolutely crucial because that, to a large degree, modulate the predator-prey interaction, which is the primary way that matter gets transformed up the food chain.
And I'm going to jump in and pretend I'm on the panel. So my response to that one is that I think DVM is sort of a second order trait. So metabolism, I think, is something that's really tightly coupled to the physics, whereas DVM is a choice. And so therefore, there's going to be other secondary traits that are going to drive those sorts of things. And that's where, as a physiologist, size for me is that obnoxious covariate, and I want to think about those things. And I think you get light, and then I think you get predator field, and then you think, oh crap, am I going to survive a migration up and down because I don't have enough energy? And I think we can get to those. We don't throw out the baby with the bathwater. Size still matters, and actually evolutionary history matters. Are you a deep species, or are you a surface species? And can you outcompete myelinated copepods? So.
It sounds like what you just described is migration length scales with the predator length.
Right. And I would say that in terms of the large-scale biogeochemistry, that migration may not be very important over most of the world, but it may be actually quite important in getting better oxygen minimum zones. Because right now we tend to, if we do what we do in most of the models, which is reduce remineralization rates within the oxygen minimum zones, they tend to spread downwards vertically. Whereas if you allow the ecosystem to compress, my instinct is that detritivory is going to be really important in actually spreading those oxygen minimum zones upwards. So instead of having a positive feedback where they get out of whack, you put a negative feedback in the model where it's easier to get them right.
I also think you can probably use traits to describe what the most important environmental factor might be. So in the example of DVM, I guess, if you have the ability to see, if sight is a trait that you have, then that's going to change your reaction to light versus if you can't see, you're relying on chemistry to tell you if there's predators around. So those traits are going to modify your behavior.
Susanne Mendendoyer, University of Rhode Island. Getting back to the idea of do we need these allometric relationships in the future, I think something that would be really helpful is to remember they're log-log relationships, and they're really excellent to give you constraints. The minimum value ever observed is this, and the maximum value is that, rather than to say, here's the line, and I'm going to read off the x-axis what my y value is going to be. So, and I think that will be a very important tool.
I agree, and if I can comment on that, because if we look at this relationship, I mean, the scatter is actually enormous, right? At least a factor of 10, sometimes more. So they work well when you look at the big scales, but when you look at small differences, they're small. But what I think of the idea of adding the additional traits is to begin to explain the variance across those with other explanatory parameters, essentially.
Yeah, I think it's really common to see when you're looking at, like, I'm thinking back to the 2015 paper I had up there where I was modeling fecal pellet production rate as a function of size. And it was exactly what you're describing, where you had a line that constrained all the other data points below it. And I think that's a pretty common type of plot in these relationships. So I agree with that.
Sure. Matthias, UBC Canada. I am a huge fan of Longhurst provinces, and this is I've been holding the question since the first presentation. In the context of allometry, do you think we should make it more systematic? Because Longhurst provinces are a bit coarse on the boundaries, and I think that moving forward with a system that would make ecoregions big and then break them down systematically and consistently all the way down to the local scale would be super useful for modeling and capture big things and small things.
Yeah, the stuff that Willie did is based off of basically trophic state, so eutrophic, mesotrophic, oligotrophic, which goes into the Longhurst provinces. So I think that the thing that's nice about the Longhurst provinces is they're just they're easy boxes to define. I think another question though is maybe better, or a better question or similar to that is what's the connectivity between those boxes? How is information or a signal propagating through those boxes, between neighboring boxes, and maybe some set of nested biogeographic provinces that take into account that connectivity might might be another way to go, or another way to think about the way that the global ocean works, or its connectivity sort of across places.
I'd like to throw a wrench in the machine and say that there's another way to think about the ocean provinces that I've been talking to people about, which is thinking about where energy is stored from year to year. So phytoplankton don't store energy from one year to the next. They store energy temporarily, like they're turning over really fast. And I'm super biased to the north of, I guess, the subarctic North Atlantic. That's my point of reference. And you could think about there are certain species of copepods that are responsible for carrying huge amounts of energy from one year to the next. And so you could actually redefine the ocean in terms of who stores energy. Like we think about, I guess I'm thinking on land, biomes on land are defined by tree types, and that's related to precipitation and this and that, but they're responsible for capturing energy and shuttling it between years. And so, I mean, in the North Atlantic that I think about, Calanus fumarchicus is that species that takes advantage of the seasonal cycle Longhurst described and takes that energy and makes it last until the next year. And that's really important to the functioning of that ecosystem. So I'm just going to put that out there as a thought of maybe a new way to think about defining biomes in the ocean.
But then we would often have to think about fish.
Yeah.
I'm glad you said that, Amy. That's a good segue to my question. So Debbie Steinberg, Virginia Institute of Marine Science. So, one thing I've been thinking about as I've been listening to all your talks is that even with size as a master trait, there are a lot of organisms that we don't even have good information on their feeding or respiration rates, like in certain places, like in the mesopelagic zone, or for certain organisms like gelatinous zooplankton or fish. And mortality, we don't, that's a really hard one for zooplankton ecologists. So in thinking about, you mentioned that the plant community is having this discussion about which traits should they use, and I think it's so much easier for them. And I'm just wondering, what do you think, if size obviously is a really important one, I'm thinking about, well, what's the next one that we actually can measure or that we know? And there's a lot of choices, and I think a lot of it depends on the question you're asking. So can we as a community really come up with, here's our 4 traits? And I understand why you want to have it consistent, because we want to compare across the world's oceans, but can we really? That's a hard problem for us oceanographers.
So you're asking, can we really do that? I agree that it depends on the question. And Karen, for instance, looked at a specific question and with a specific set of species and looked at some traits that exactly is related to that. So what I try to say, if you think about the question is describing global structure and function of the global ocean, and particularly how it changes, which traits would it then be we should describe? I think that with the investment in phototrophy, that would be one of my go-to choices for that, at least for the unicellular plankton. And then something that relates to, it's all about relating to how they capture energy and how they, so that would be the other one. So that's that photo investment or the diastrophy, right? That's about how they get nutrients or light. And the other one would be how they defend themselves against predation, which is very much a vacuole. It's partly a defense trait. By being larger, you are less exposed to predation, or by having the shell, you are less exposed to predation as well. So these are the things that modulate mortality, as you mentioned, and the uptake of resources, which are the 2 main things that matters.
I think in relation to land, part of the challenge for us is that we're in 3 dimensions, right? And so I agree, like the depth question is really important. And maybe when we're thinking about defining biomes, do we actually have 2 sets or 3 sets of biomes relating to depth? I mean, can we actually look at a whole water column and assign traits to that whole water column, or do we have to break it up? It's a good question. And
I would also just say that one of the things that I find attractive about thinking about allometry is that we could, we had that, the plots I showed at the end there were for all microzooplankton versus particle-based nanoplankton from space, right? You could imagine doing that with more functional groups and at least starting with the stock behavior, right? I agree with Amy that of course we want rates, but if we start with how stocks relate to each other, maybe that gives us some clues. And that is at least an easier thing because you could do that with imaging now in ways that you couldn't before.
Okay, well, maybe that's fate. Yeah, I won't ask.
Okay. Okay.